Good afternoon, everyone. Sorry about that. My name is Patty Ingham, and I'm the director of the Institute for Advanced Study here on the Bloomington campus. On behalf of the staff and faculty affiliated with the Institute, I'm especially pleased and proud to host this year's second Branigin lecture on the Bloomington campus. The IAS, for those of you that don't know about us, is a unique institution. It's the only interdisciplinary research center on campus not associated with a single or specific focus or topic. Instead, this constitutes one of our greatest strengths. Dedicated to the broadest kind of radical interdisciplinarity and research collaborations, we at the IAS are committed to promoting rigorous and nimble engagements, working with campus faculty researchers, with our extraordinary repository and museum partners, with our residential and visiting fellows and invited guests. We aim to foster conversations capable of examining current issues, contextualizing urgent cultural questions. This year marks the 40th anniversary of the Institute, an unusually long era for local institutes like ours, and indeed, IAS is one of a very few interdisciplinary institutes of its type hosted on the campus of a public university. This history means that as we mark our anniversary, we call to mind the fact that we are standing on the shoulders of giants, all those past IAS leaders and staff members, and the faculty who, for over 4 decades, have brought intellectual leaders of the highest caliber to the campus. We are delighted to welcome this year's spring Branigin lecturer, Professor Randy Schekman, whose personal generosity and kind humility has been evident to those of us at the Institute over the past months of our planning. Professor Schekman's work reminds us in sharp ways the power of research collaborations prompted by, rather than opposed to, basic research and basic science. These questions and methods continue to be urgent, both for the future of the university and for the publics we serve. In a moment, I will turn the podium over to distinguished Professor David Allison, Dean of the Indiana University School of Public Health, who will introduce Professor Schekman. But before I do, I wish to acknowledge those who have made this lecture and this visit possible. First and foremost, the staff of the IAS, Associate Director Suzanne Godby-Inglesby, Coordinator Elizabeth Kang, and Graduate Assistant Taylor Zartman, with the able assistance of Alex Teschmacher of the College Arts and Humanities Institute. And thanks, of course, to Dean Allison and to Justin Otten at the School of Public Health for our help with matters large and small. To Dean Rick Van Kooten in the College of Arts and Sciences and the Departments of Chemistry and Biology, especially distinguished Professor and Lilly alumni Professor David Giedrock, who guided some of our planning and facilitated our initial introductions with Randy. Resources and support from the Vice President's Office of Research and the research staff on the Bloomington campus have also proved crucial, particularly Vice Provost for Research Brea Perry, Ed Dallas-Comatale, and Sherry Knighton-Schwand. Last, but most importantly, this afternoon's lecture has been made possible through a generous endowment of IU alum Jean Lois Portreras Branigin. Dean Allison.
Well, it's truly a privilege to be here today, and I appreciate the remark about my being a distinguished professor, which is code for has gray hair. That's how you know you're distinguished. It is really a great privilege to be here with someone I admire as a scholar, as a human being, as a friend and a colleague, Professor Randy Schekman. Professor Schekman is a professor at Berkeley University. He is, as many people I'm sure know, a Nobel laureate, and he won the Nobel for his work in cellular biology. He has been the editor of the Proceedings of the National Academy of Sciences. He's a member of the National Academy of Sciences. He founded eLife, a prominent journal that is pushing boundaries of the way we do publishing to think about new models, whether they're pushing them well or not. We can all argue, but that's part of pushing. Push forward new ideas that we can argue about in human spirit. I can tell you a little about the science. I can tell you about the scientist. I can tell you a little about the man. I'm not going to tell you much about the science, because really, I'm not a cell biologist, and I probably wouldn't get it right. And he can tell you more about science better than I can. I can tell you there are many papers and many grants and many achievements. I want to tell you a little bit about the scientist. So we heard about Newton a moment ago. Or really, we heard about the phrase, standing on the shoulders of giants. It was Newton who said that. And Newton was an interesting character. Newton was a great scientist in addition to doing some great science. Newton, during the pandemic of his time, was sent home from his university. And during that time, came up with the universal law of gravitation. And shortly thereafter, invented the calculus, made leading contributions to optics, and so on. So that's our friend Newton, really amazing guy. But he was much more as a scientist than just his science. He also became president of the Royal Society. He served society as chairman of the Exchequer at one point. So he was more than just one scientific project. He was a scientist with a broad view, just as Professor Schekman is a scientist with a broad view, who did his seminal work in cellular biology, but then went on to say, it's time for me to give back, and has given back, and not only thought about the science of health, but has thought about the health of science, and has established and leads a foundation funded by philanthropy, which I'm sure is going to tell you about. He is literally giving back to others. He gives back through extraordinary mentorship. He gives back for having been the editor of PNAS. He gives back for poking at the field in a very appropriate way about rigor, reproducibility, and transparency, and how our publishing models may not be optimally serving us to promote the best science, the most equitable science. And so he's giving back in a wonderful way. Now, unlike Newton, Randy Schekman's a good guy. Newton was not a very nice person. As chairman of the Exchequer, he sent settlement to the gallows. He was bitter. He took pride in having broken Leibitz's heart over the battle of who invented the calculus. So he was involved in things beyond the science, but not in the development of things. And I want to say, Randy's not, if you'll forgive me, he's not a shy, retiring pussycat. But he knows when to punch up and not to punch down. When I hear Randy talk about mentorees and students and postdocs and early career faculty, and quite frankly, even crazy deans out here in Indiana who don't quite know what they're doing and need a little help, he helps. He reaches out. He's generous. He's in a very generous spirit. But when you get him in with a more powerful person who he feels is not doing the right thing, Randy's a street fighter. He's ready to throw some punches. He's not a shy, retiring guy, and that's discredit. He's ready to duke it out, and not just in the interest of peace and harmony, say we'll accept anything less than the best for our people, because he's concerned about the health of science, and not just the science of health. So I have been the recipient of his largesse. He's a wonderful man. I consider us very lucky to have him here. Randy, welcome.
Thank you very much, Patricia and David, for those two kind remarks. I'm going to give two lectures. The first lecture is the lecture today concerns a kind of extracurricular activity of mine that has grown to a very large interactive, collaborative research network dealing with Parkinson's disease. And tomorrow I'll talk about the research in my own laboratory on vesicular traffic. The two subjects relate, as it turns out, because some of the principal features that we now understand about Parkinson's disease have to do with genes and thus of proteins that are involved in certain aspects of membrane traffic within human cells, particularly within the nerve cells of the brain that coordinate to produce the neurotransmitter dopamine, and which interact with the upper regions of the brain that control aspects of movement. So I'm going to this afternoon give you a lecture in several parts. The first has to do with the nature of the disease, the pandemic, the efforts to control it, what really little we know by way of molecular or cellular or tissue aspects of the disease, some very promising leads. I'll tell you about my own personal commitment to the understanding the disease, and then I'll tell you about the effort that was given to me, an opportunity, really a gift, to try to organize an international consortium of investigators. So let's begin with a description of what we know about the disease. These first slides are unfortunately in a time sequence. So Parkinson's is in a real way a pandemic. It is growing in incidence more rapidly. It's the second most prevalent neurodegenerative disease that afflicts people today. It's growing more rapidly than Alzheimer's disease due to a lot of influences, some likely environmental, some genetic, and certainly some of familial origin. It's also a difficult disease to clinically diagnose, and so its incidence is rising, possibly because of better diagnostic tools. It is growing in incidence around the world. It will soon take over all of the world. It really has no boundaries. Over half of the new cases likely to emerge in the next decade will come from China, so there's an enormous interest in research in this area in mainland China. The disease is not really a single disease. It's likely a spectrum disorder. It presents in very different ways. There are some patients who develop the disease very early. They progress very rapidly. Their survival is low. They develop dementia. Approximately 30% of the patients who initially present with symptoms of Parkinson's succumb to a form of dementia that's distinct from the dementia that afflicts people with Alzheimer's disease. There are others who survive a very long time. Michael J. Fox, for instance, perhaps the best known victim of Parkinson's in this country, has suffered for almost 40 years with the disease, and he's now very slowly coming to the end of his life, in spite of having fought all these years. In his case and in others, there's little evidence of dementia. The problem with viewing it as a single disease, as neurologists have done for almost 200 years, is that it's been a failure to try to develop drugs that might be useful in treating all patients. There are some drugs that are effective in mitigating some of the symptoms. For instance, most patients who present with Parkinson's are used with a form of dopamine replacement that has access to the brain and which can control the awkward movements that are associated with the early forms of the disease. These patients can live normal lives for several years, but inevitably, possibly partly in relation to the treatment of the drug, they develop dyskinetic movements, this very awkward movement that is often associated with the progression of the disease. And underlying this is a continuing progression of the disease. The arc of the disease has not been mitigated by any of the drug treatments. More recently, a surgical procedure called deep brain stimulation was developed by an American and an Algerian French scientist. It involves implanting electrodes bilaterally into a region of the brain called the subthalamic nucleus that controls awkward movements associated with progression of the disease. And with mild electrical stimulation elicited from a battery pack that's implanted in the shoulder of a patient, these movements, these awkward movements, can often be controlled for a very long time. So it really does help in the symptoms, but it does not affect the progression of the disease. It is inevitable and inexorable and essentially leads to death. Now let's focus on what we know at a cellular level and even at a molecular level. It's known that the disease is focused on a region of the brain called the substantia nigra, which is a collection of some tens of thousands of cells in the midbrain that are organized to produce and secrete dopamine and which innervate higher regions of the cortex that I'll tell you about later. Here is a section of a brain taken from a normal person who died for other reasons, the substantia nigra can be recognized because dopamine, a product of dopamine, is melanin, which is deposited in these cells and which gives them a natural coloration that can be visualized in brain sections. So it's a convenient marker, of course, only on a biopsy patient. And patients who have, in this case, one died of Parkinson's, this melanin accumulation is drastically reduced as a result of the progressive death of dopaminergic neurons. These neurons are extraordinarily active. I'll show you an image of a dopaminergic neuron later. They are always on, they're always transmitting signals, they are hyperactivated, and so they're very vulnerable. And in normal patients, these cells are inevitably dying at a slow rate. And so many people who progress into their 80s or 90s develop symptoms of Parkinson's disease simply because of the normal progression of death of these cells. Patients who suffer from Parkinson's, however, these cells die at a greater rate, and patients present with symptoms. By the time they presented with physical manifestation, over half of the dopaminergic neurons have died in the course of the decades before the disease is represented. Now, about 100 years ago, a British physician named Frederick Louis, on inspecting biopsies of sections of patients with Parkinson's, noted that these tissues often are accompanied by inclusions within dopaminergic neurons of quite substantial deposits, it turns out, of fibrillar material and protein aggregates. These have been called Lewy bodies in honor of Louis' discovery. The basic constituent of these Lewy bodies is a protein called alpha-synuclein, which is a small protein that's found in the brain, found specifically on synaptic vesicles. Its normal function is really not entirely clear. But when this protein begins to aggregate, either for reasons of mutation, certain alleles and familial forms of the disease produce a form of the protein that begins to aggregate, it's believed that these aggregates are somehow related to the disease. It's not certain that the Lewy bodies are themselves the reason for death of dopaminergic neurons, but it is a correlation. Correlation, as I learned as an undergraduate, is an agent of the devil, so we can't really assume that these Lewy bodies are the basis of the disease, but they certainly are a measure of it, and what we would dearly love is to have a non-invasive probe, a PET scan probe of Lewy bodies that would allow patients to be evaluated during the course of their life, as can be done with amyloid plaques in the brains of Alzheimer's patients. But thus far it has not been possible to develop a sensitive PET scan probe of these Lewy bodies, so the only way that we have of evaluating these is by biopsy of patients who have succumbed from the disease. Very famous and tragic example of this was the comedian Robin Williams, whom you may recall committed suicide. It was as a result of an enormously rapidly progressing form of Parkinson's disease that was recognized just months before he took his own life. His wife, who was struggling to cope with this, commissioned a complete evaluation, section analysis of his brain after he died, and they discovered Lewy bodies all over the brain, not just in the substantia nigra, but also in the hippocampus, which may have explained his very erratic behavior before he took his own life. So there's definitely a correlation. We would love to know if it is a meaningful correlation that could be the basis of some treatment, and there's a great deal of effort underway to try to control this protein alpha-synuclein by way of hoping to control the proliferation of these Lewy bodies. Now, in the course of evaluating lots of patients, a German, excuse me, that's it, before I segue to that, it's possible actually in the laboratory to recapitulate the formation of these Lewy bodies in cultured neurons. And here's an experiment done by a scientist in Lausanne, Switzerland, with a form that was where these cortical neurons from a laboratory animal were cultured and transfected with a gene to allow the overexpression of alpha-synuclein. And after a couple of weeks of culturing, these cells, by immunofluorescence, show a quite substantial aggregate of alpha-synuclein and other things occupying a quite sizable fraction of this neuron. And in thin section analysis, this is shown in false color. The structure is seen to contain many other things besides protein aggregates, organelles, fibrillar proteins, quite a mess, so large as likely to evade the normal cellular machinery that would be mounted to try to dispose of these by degradation or by a process of engulfment within the organelle called the lysosome. So this is part of the problem. Now, in the course of sections of many patients who succumb from Parkinson's disease, a German physician, Dr. Brock, devised a model, really just a speculative model, that suggests that there may be some progression of Lewy bodies from the brainstem, possibly via the vagus nerve from the gut into the lower regions of the brain. And it's quite interesting to note that Lewy bodies can be detected in other tissues, not just in the brain. They can be seen in the nasal mucosa, which may explain why some patients who eventually present with Parkinson's disease develop a poor sense of smell. It can also be detected in the gut, where patients, again, who present eventually with Parkinson's sometimes have digestive problems and reflecting what would in many cases just be a simple explanation, but which eventually turns out to be because likely of Parkinson's disease. And it's been noted clinically that there may be a progression of these Lewy bodies from the gut up the vagus nerve into the brain stem. And a Danish study some years ago suggested some evidence for this because people used to be treated for ulcers by having a vagotomy, a resection of the vagus nerve. And it was noted in thousands of patients who had this surgical procedure that there was a much lower incidence of Parkinson's disease later in life. This is not something that you would do prophylactically, but it's certainly an interesting suggestion that the disease may progress from sources outside of the brain. Indeed, it's possible to take an aggregated form of alpha-synuclein and inject it into the gut of a laboratory animal and observe over time the movement of these particles up through the vagus nerve into the brain, throughout the brain. And so there's a great deal of interest in how this protein and its aggregate forms may move within tissues from cell to cell, eventually progressing into the brain. And as Brock suggested, by a staging approach, that it's possible to evaluate the influence of this progression as the disease develops, from olfactory disturbances, such as I've suggested may often occur long before the movement disorder, to disturbances of sleep and motor dysfunction. I'll tell you about an example of sleep disorder later on, and then finally some cognition defects as the disease progresses. All right, now I'm going to delve into a little more cell biology to explain some of the subcellular organelles that have been implicated in Parkinson's disease from the genes that we know are in familial forms. There are now some 20 different genes, mutant alleles of which can be found in family forms of the disease, where there's an inheritance and a very high penetrance of this mutation in subsequent generations. One of the particular organelles of great importance in the progression of Parkinson's disease is the mitochondria. So please excuse me for those of you who already know about this, but I'm going to just tell you a little bit about the mitochondria and why it is so critical, not only to normal cell function, but how its surveillance and how damaged mitochondria are removed, that is also extremely important in the survival of dopaminergic neurons. So here's the mitochondria, and it probably originated by an endosymbiotic relationship with an early gram-negative bacteria. It has two membranes, an outer membrane and an inner membrane, and a matrix interior. This can be seen in cross-section in an electron microscope image, where the infoldings of the inner membrane organize a process called oxidative phosphorylation and electron transport, which is intimately involved in all of our cells in producing the vast quantities of ATP that we need to to actually do the chemistry of life. Now, the organelle is enormously complicated. It receives almost all of its proteins coded by genes in the nucleus that have to be made inside the cell and then delivered into the interior and to assemble and function in the mitochondria. The organelle also has its own genome, a small DNA molecule that at this point in evolution only encodes for a few genes, and these genes code for proteins that are engaged in the aspects of this electron transport chain that is used to power the production of ATP, the energy currency of the cell. So there are two things about the functioning intact organelle that must be surveyed to make sure that the organelle is intact. One is the cell goes out of its way to make sure that the DNA that's in a mitochondria stays in the mitochondria. Anytime a mitochondrion is damaged and its DNA spills out into the cytoplasm, that sets in train a death response pathway that eventually kills the cell. The cell would rather kill itself than have the mitochondrial genome exposed because of the consequences of mitochondrial damage. The other thing is that at the end of the electron transport chain, there's an oxygen radical that is held in, at the end of this long series of events that must be held in check to avoid escape. And if the mitochondrion is ruptured, these oxygen-free radicals can escape and enter the rest of the cell and wreak havoc in the nucleus by breaking chromosomes in the nucleus. So the cell goes to elaborate means to keep this organelle happy and to get rid of it when it's not doing its right thing, its proper thing. Now, what I want to tell you about is how the cell manages to get rid of damaged mitochondria. There's a process that cells have evolved from microorganisms to us called autophagy, or self-eating. It's actually a very important pathway in the cell because cells often have to turn things over to make new amino acids and sugars and nucleotides to mobilize new pathways. And so the cell will dispose of organelles or dispose of particles just in order to salvage amino acids to survive. And the same thing, the same pathway is mobilized to get rid of damaged organelles. It does so by a process of autophagy, a general term, or mitophagy, specifically referring to the destruction of mitochondria. It does so by, as you'll see in a moment, when an organelle is damaged, it gets a special tag from a cytoplasmic machinery, and this tag is recognized as an eat-me signal or a destruction signal. And that tag stimulates the envelopment of the damaged mitochondria or other damaged organelles. These organelles become enveloped by a new membrane in the cell called a phagophore. And the phagophore membrane is itself joined to another organelle in the cell called a lysosome. The lysosome is kind of the digestive tract of a cell. It's filled with enzymes that ordinarily would be noxious, but in the environment of the lysosome, these enzymes can chew up anything. They can chew up nucleic acids, fats, sugars, proteins, and recycle their constituents into the amino acids and sugars that a cell would use. And these nutrients can then be spit back into the cytoplasm to be reused for other things. So the lysosome, though it's a destructive organelle, serves a very important function. And in the context of damaged mitochondria, it serves an essential function to get rid of them, to destroy them so that they won't do the kind of damage that I alluded to. All right, now why am I telling you this? Well, it turns out that two of the most interesting, if you're a scientist, interesting genes that afflict certain familial forms of Parkinson's are genes that encode the cellular machinery that puts a tag, an eat-me tag, on a damaged mitochondria. These two genes have been given the cute names of PINK1 and Parkin, and they encode enzymes that put a little peptide tag on a damaged mitochondria. It becomes decorated with this little peptide tag, and these tags then cause the envelopment of the damaged organelle by a phagophore, and thus finally degradation in the lysosome. So there are a number of investigators around the world. One whom I have been in touch for some of these slides is an investigator at the NIH by the name of Richard Ewell, who's made a beautiful research career studying these PINK1 and Parkin genes. And this is just a quick summary of what he's found. As I said, the mitochondrion exists normally, functions when it's healthy, but if you've damaged the mitochondrion, and I'll show you an experiment where we can actually damage the mitochondrion in cells grown in the laboratory, it causes, the damaged mitochondria causes these two proteins, pink one and Parkin, to act together to put a little tag on the mitochondrion, and that little tag then is recognized by the phagophore membrane, which swallows it up and dumps it into the lysosome. So let's look at an actual experiment. This is a way of visualizing cells using special probes, antibody molecules, to which you can covalently attach a chemical, a fluorescent chemical that will allow you to visualize these cells in something called a fluorescence microscope. So here's a cell, a resting cell, and in this resting cell, for which they've not been perturbed in any way, their mitochondria are healthy and making ATP and not being turned over. If you look at the tag, the protein that would eventually tag damaged mitochondria, it really isn't. It's so diffuse over the entire cell, you can't even see it. It's just all over the place, waiting to be used. And when you align that with the appearance of Parkin, which is also all over the cell, it's not organized in any way, there's no particular organization in this undamaged cell. Now, if in another experiment you take these cells and you treat them with an agent that causes the electron transport chain in the mitochondria to be disrupted, the cell says, aha, there's damage here, we better get rid of the mitochondria. Of course, that would be toxic, but in this experiment, you can use that condition to visualize the reorganization of this protein Parkin and of this tag, ubiquitin, aligned onto mitochondria, which with time eventually will be swallowed up by the lysosome and degraded. So, the lesson from Richard Ewell and other laboratories is that these two genes, one can explain why Parkinson patients suffer. It's because their dopaminergic neurons, which for reasons that I'll show you in a moment, are so vulnerable. Whenever they lose the ability to remove damaged mitochondria, they suffer mightily and they die as a result of a failure in quality control in the destruction of mitochondria. When cells have damaged mitochondria persist, they will inexorably be propelled into a pathway of death. And so that really could explain why these cells in the brain are particularly vulnerable. But I hope to convince you of that by showing you what a dopaminergic neuron actually looks like. All right, now one other observation, which is actually brand new and which has been supported by this network that I'll tell you about, affects a different pathway. It also affects dopaminergic neurons, but in a fascinating, quite indirect manner. So let me explain that to you. So here's a simple depiction of a dopaminergic neuron. It's far from the real thing, but just for the time being. Dopaminergic neurons communicate in the brain over a very long distance with, it turns out, with millions of other cells. And among them are special cholinergic interneurons in a region of the brain called the striatum. Now, the way they communicate is through a curious pathway that has the name hedgehog signaling. For those biologists in the audience who know about this, for the rest of you, it's just a funny way of describing a form of chemical communication between two cells. The communication involves a signal emanating from the dopaminergic neuron that is picked up by a kind of an antenna on this cell, a cholinergic interneuron. This antenna is a little piece of membrane that protrudes from the cell that's called a cilium. It's a primary cilium, a sensory cilium, and it organizes, the sensory cilium organizes receptors that respond to signals coming from other cells. And when this signaling pathway is functional, the cholinergic interneuron cilium recognizes a signal emanating from the dopamine neuron, and it elicits a path of production of a growth factor called GDNF that is secreted by the cholinergic neuron, diffuses back to the dopaminergic neuron and nourishes that cell and allows it to flourish and continue to function. If this pathway of communication is interrupted in any way, the cholinergic neuron will not survive, nor will the dopaminergic neuron survive. Now here's an amazing news story that's only just been published in the last couple of years. Because it turns out that one of the genes that's implicated in genetic forms of Parkinson's, a gene called LRRK2, LRRK2, which encodes an enzyme called a protein kinase. It's an enzyme that will take a phosphate from ATP and covalently attach it to a target protein. It turns out that mutations in LRRK2, and this is really quite surprising, mutations in LRRK2 that cause Parkinson's actually turn on the LRRK2 kinase. Its activity as an enzyme is elevated. Usually you think of mutations damaging a protein, lessening its activity. In this case, it's a dominant mutation that activates the enzyme. And when it's more active, it improperly attaches a phosphate to another protein called a RAB protein. And this particular RAB protein, when it has a phosphate on it, suppresses the production of primary cilia in the cholinergic neuron. These discoveries were made by a team of investigators, one in Dundee, Scotland, and one at Stanford, who are prominent in our network, and I'll tell you about them later. And they've shown very recently that they can see this actually in biopsy patients who have suffered from mutations in LRRK2 or in associated genes, and even those who come with non-genetic sporadic forms of the disease, that the cholinergic interneurons in the striatum in biopsy patients who died of Parkinson's have a dearth of primary cilia, which may be directly tied to the inability of a functional hedgehog signaling pathway between these two cells. So this is very exciting because it suggests some very obvious clinical possibilities to try to correct this defect, which I have to tell you about later if you're interested. All right, now I'm going to turn a little bit because I want to emphasize again why dopaminergic neurons are so vulnerable. This is a cartoon of a typical nerve cell. It's nothing like you'd find in the brain. It's much more crowded in the brain. But when you think of a nerve cell, you think of a cell body that has a nucleus and mitochondria, and you may think of dendrites that project from one side of the nerve cell, or an axon, and together with nerve terminals that project from the other. This is the classic image of a nerve cell in the brain. In contrast, have a look at what a dopaminergic neuron looks like. This is an amazing cell. It's amazing that it even survives to do what it does. Here is the cell body. Here are dendrites that project away from the cell body. Here is the axon. And instead of just a few nerve terminals that connect to a few cells, dopaminergic neurons have as many as 1 million nerve terminals, each making a different contact with regions of the striatum and the cortex of the brain. So, a brain has 12 billion nerve cells. The typical nerve cell makes maybe 1,000 connections with other cells, so there are 1 trillion possible connections. But dopaminergic neurons are even more involved in their interaction with movements of the brain that control executive function and movement. So it's little surprise that these cells are working overtime and are poised at the edge of vulnerability. Any damage to a dopaminergic neuron makes it less likely to survive over the length of a human brain. And that's almost certainly why mutations in genes like LRRK2 or PINK1 and Parkin, which you'd think would affect all of the cells in our body because the pathways they control affect many of the cells in our body. But these mutations seem to have as their principal effect on the dopaminergic neurons, and that is because it is simply so vulnerable. Okay, well, now I have to turn to something that I'm not used to doing, which is to describe my personal path, why with a basic scientist who spent much of my career working on vesicular traffic in Saccharomyces cerevisiae, I'd be interested in Parkinson's disease. And I'd rather not have to tell you this story, but I have a personal stake in this. My wife of now almost would have been 50 years died of Parkinson's 5.5 years ago. We had a wonderful life together. Here we are at our wedding nearly 50 years ago at a park outside of Stanford. We noticed actually early on after we were married that she had a poorly developed sense of smell, but you know, who makes anything of that? There are lots of reasons to have a poorly developed sense of smell. We never made anything of it until she presented with symptoms of Parkinson's. Initially, they were quite casual symptoms. She was sort of had aches and pains, and at one point she had a frozen shoulder, likely because of inadequate arm swinging movement, which often, I'm told, subsequently is often what happens initially with when Parkinson's presents with clinical symptoms. But the thing that really, at age 48, made it very clear was when she went to the neurologist and said, what could it be, he didn't have an answer for her, what her ailments were. And she reported then on a second visit to the neurologist and showed me one evening an amazing demonstration where she tried to sign her name, and it was in the tiniest print. I couldn't imagine that she could even do this. But it turns out that's a classic symptom. It's called micrographia. It's a loss of fine motor movement rather than a gain of fine motor movement. And so she said, well, what could this be? And he said, it could be Parkinson's. And she said, well, what could I do? How will I know? And he said, well, you could go on a course of the drug that's a dopamine replacement. And I'll never forget the call that I had in my office that afternoon when 2 hours after having just taken one pill, all of the symptoms melted away. So that was the beginning of a long 20-year torturous path. For the first years after she was diagnosed, she was very happy. She could be treated with this dopamine replacement for 5 years. Most people didn't even know she was ill. You really couldn't tell. But inevitably, as time went on, she developed the awkward movements that are associated with progression of the disease. These didn't bother her. They were more uncomfortable for other people who were found it awkward to be around her. But she thought it was great because it was good physical exercise, and she was able to contain her weight by these awkward movements. But that was, of course, only the beginning. She progressed to more serious forms of the disease with aches and pains, and her neurologist suggested the procedure that I told you about called deep brain stimulation. So she had the surgery. It went very well. She was able to control the voltage, and most of her symptoms just melted away. But the disease progressed inexorably, and nearly 6 years before she died, she showed signs of dementia. Here we are at our house up the coast, and here's my son on the left, a former music student here at IU. Was still relatively happy. We could still have a normal life. The kids were grown and gone at that point, but the progression of dementia was really the worst of it because she lost her ability eventually to communicate. Not because of any mechanical problem, but because she simply couldn't remember what she was saying. She would turn to me and start saying something, and then her face would grow blank because she couldn't continue. When she first presented with the symptoms of dementia, she had a battery of cognitive tests, and I'll never forget the moment when the neurologist said, I'm sorry, but I have to inform you that I've called the Department of Motor Vehicles and you're no longer able to drive. That was a devastating blow. It changed our lives. We had to have care for her during the day and care for her overnight when I traveled. There still were happy moments. Now, of almost 10 years ago, she was well enough to join my family and me on our magical trip to Stockholm. And here she is with my father, who is since deceased, who was, of course, particularly happy to be there celebrating with us here we are at the main banquet in the City Hall in Stockholm. But as we got on the plane to return home, she went to the bathroom and came back and said, I was talking to somebody waiting in line, and where were we just now? What were we doing just now? I mean, literally on the plane home from Stockholm, she didn't recall where we were. And it went downhill from there. I had access to the best neurologists around the world. She had been diagnosed with a form of Parkinson's that advanced into what is called diffuse Lewy body disease, although they couldn't evaluate the presence of Lewy bodies in her brain. Her behavior was consistent with that. I had access to the best neurologists who were focused on diffuse Lewy body disease, and I said, well, I know there's nothing you can do, but perhaps you can tell me what to expect going forward. And they always had an answer, and they were always wrong. Here, as things got worse, she slept an ungodly number of hours in the morning. I had trouble giving her medications because she was almost like a zombie. And finally, at the end of her life, it became almost unmanageable. And then now, 5.5 years ago, in the middle of the night, she died, either of heart failure or of a stroke. We don't know what it was. So I come to this having suffered the frustration of a patient supporter, not knowing what to expect. The disease appeared out of nowhere, it advanced in no predictable way, and she died for reasons that no one could explain. Fortunately, near the end of her life, I was approached by the chief financial officer of the Sergey Brin Family Foundation. This fellow was a really bright guy, but not of any biological interest background. But he was, on behalf of Mr. Brin, was asking about what might be done to support more research on Parkinson's disease. It turns out, of course, as you know, Brin is enormously wealthy. He was the co-founder of Google, but he has a vested interest in Parkinson's. He has the most prominent mutation in the gene that I told you about, LRRK2, an allele that causes just a twofold elevation in its kinase activity, enough to cause progression of the disease. His mother has the same mutation. It's a dominant allele. They are both genetic carriers. His mother is ill with Parkinson's and has been for some years. Mr. Brin himself is perfectly healthy thus far, but he's obviously at risk. Brin has had an enlightened self-interest. He's invested enormous sums of money in the efforts of the Michael J. Fox Foundation to support research on Parkinson's. It is a wonderful organization, I can attest to that, but they have their primary interest in supporting families and in clinical research, and only a small effort on basic science. And Brin had decided, quite before I became involved, that what he wanted to invest in was a substantial basic research investigation, to invest a substantial sum of money in an effort to understand the molecular and cellular basis of the disease, so that new drugs might become available through such efforts. And so when my wife died, I was asked to chair a committee that had been established to begin an exploration of the areas that might benefit from a substantial infusion of cash. And I, at that point in my life, what better could I do with the rest of my career? And so I've joined forces with a very small team in Washington. We're called Aligning Science Across Parkinson's, ASAP. And we in the first 2 years, before we started funding teams, we developed a strategy that I want to share with you about what we will focus on in our effort and why we feel so strongly that this would be more than what the NIH does. Of course, the NIH supports a lot of basic science on Parkinson's disease. Most of it is given to individual investigators. Some is for team science, but it isn't really an integrated form of team science. So we wanted to develop a strategy and a mechanism for directing what potentially could be $1 billion to organize teams of investigators focused on the major questions associated with Parkinson's. Parkinson could be like a puzzle, obviously more complicated than this puzzle. We know some of the pieces, mitochondrial dysfunction or failure to remove damaged mitochondria, failure in degradation of proteins. There are elements of neuroinflammatory processes that may exacerbate the lesions in the brain. There's obviously synaptic pathology, and then there are the mutant forms of alpha-synuclein. Ideally, we would have enough basic science to understand how 1,000 pieces fit together. The 20 genes that we know of don't have any obvious connection except in certain instances, but yet they are somehow related. They all somehow end up focusing on dopaminergic neurons. We want to understand how they all fit together. So here are the themes that we devised in the course of a couple of years of meetings with neurologists and Parkinson's experts. We, of course, want to focus on the genetics and the associated biology and cell biology. You know, the LARC2, the pink one, the Parkin, what these genes normally do, why they are particularly vulnerable when mutant in dopaminergic neurons, and the other 17 genes, how do they interact? We want to know about neuroinflammatory processes that may initiate or exacerbate the disease, that is the body's own immune system or the immune-like cells of the brain that may collect in lesions and make the disease worse. We want to understand the enormously complex circuitry of dopaminergic neurons. Imagine figuring out what those million neuroterminals connect to. That's going to be a major task that's not appreciated even under normal circumstances. And then we want to understand the strange and sometimes prolonged period that precedes the development of the movement disorder. I told you about poorly developed sense of smell or gut disorders. One of the symptoms that precedes the development of the movement disorder that is most mysterious, but often something close to 80% of the time progresses to Parkinson's, is a malady called RBD or REM sleep disorder. This is where patients lose their ability to distinguish sleep and wakeful thoughts. They sometimes live out their nightmares while they're sleeping, and they sometimes are so violent that they throw themselves out of bed or they flail around and harm their patients. This is a known syndrome, and it's now possible to study these people in isolation. And within 5 years, most of them, something like 80% of them progress to Parkinson. What's the connection between this disorder and the movement disorder? There must be something obviously deeply involved. We'd love to be able to follow patients by some means other than what a neurological evaluation provides. We need a biomarker such as we have with cancer or heart disease. And just very recently, within the past year, there is now a very likely biomarker that is to be able to evaluate a fluid taken by spinal tap for the presence of a seed of alpha-synuclein that stimulates the aggregation of alpha-synuclein. This has been turned into a reliable routine clinical test, and there's a very high correlation between the presence of this seeding activity in CSF and the disease. So this is a very hopeful sign that will allow patients to be monitored, if not to be treated just yet. Now, very specifically, and coming to the end, I and my partner in this, Timothy Riley, have a very strong feeling about team science and the value of team science as opposed to the usual investigator-initiated grant that most of us have lived on for our careers. We want to identify teams, or our aspiration has been to identify teams who have, in their history, before they came to us for funding, in their history, developed an understanding by collaborating on an important problem with some other investigator, either relevant to Parkinson's disease or not. Because we know from experience that some people really do understand the give and take of collaboration and are not always worried about where their name goes on a paper or where the paper is going to be published. We want people who understand that giving in collaboration is what builds momentum in science. More than the parts, it builds momentum. And so we put out a call over a period of a few years for teams, 3 to 5 investigators, at least at 2 different institutions, teams that include one early career investigator, teams that were good, that had a good gender balance, teams that brought in people from outside of Parkinson's, and where there was evidence through collaboration, publication, organizing meetings together, writing grants together, that they knew what it meant to have a give and take. We got lots of applications from superstar scientists, many of whom didn't get funding because they hadn't proven to us that they really understood what a collaboration meant. So teams, diverse talent, driving a focus on the 4 themes that I told you about. And of particular importance to me was embracing the value of openness. All of our teams have to communicate with each other online in a network that we've created that was perfectly active throughout the entire pandemic. The teams have to present in group meeting format with new results, not just published results. The teams have to all agree to sign a CDA so that they're held in confidence for new information that's shared on the network. They have to share reagents with each other and protocols with each other. And we've seen a number of new collaborations emerge from people that never met each other. They have to be willing to post their work on the bioRxiv or the medRxiv as soon as it's ready to be published, and they have to publish in an open access forum, any of the journals that offer that. So with these values, actually, how am I doing on time? Okay, let me jump into just a few things, and I'm going to have to go back online to get this next thing for you, if I can. Go online because there's an amazing little movie that I want to tell you about that one of our teams is using. Okay, all right. Okay, so one of the teams, a team led by a scientist at the University of Pittsburgh, Dr. Strick, is studying the neuronal circuits in mice that can be ablated, the dopaminergic neurons that can be ablated by the application of a drug that is neurotoxic specifically for dopaminergic neurons. And he and his team have found that the normal network that makes connections with dopaminergic neurons is what he refers to as a closed loop network. He's discovered that there's an alternative path that bypasses the dopaminergic neurons, which he's called an open loop, which he thinks, and we hope, may explain a very curious phenomenon that it often accompanies the behavior of Parkinson's disease, who have a very severe form of movement disorder. And here's an example of a young man who developed a form of Parkinson's from a genetic form of the disease that I didn't tell you about, but who's a very prominent blogger. And so let's see if, oh my, it's not showing. Damn. How do I get this on the screen here? I see it on my screen, but not on the screen up here. I should have tested this out beforehand. I had it on my screen, but it's not projecting here. Ah, that's it. Fantastic. Okay, thank you very much. Okay. So, pay attention to this guy. His name is Ben Stetcher. He's a 30-something young man who's recently had deep brain stimulation, but it doesn't really suppress the movement aspect of his disease. So here's Ben doing his usual gait, unable to walk. He's got a very serious tremor that isn't mitigated by the DBS in his case. But you give him a basketball, and it's like he was faking it. This is not an uncommon phenomenon. There is obviously some alternative circuit that controls movement, that he can elicit through some mental activity that completely suppresses the appearance of the movement disorder. And in the other image that I will not bother you with, but this is a famous case of a guy, a Dutch patient, who has a very serious movement disorder, worse than Ben's, who basically can't even walk. He falls every time he starts to walk. You put him on a bicycle and he rides around a parking lot like there was nothing wrong with him. So what explains this divergence of behavior, and can we identify the neural circuit that these folks are able to elicit, and can we stimulate the engagement of that circuit as an alternative for normal patients who then at least have a much reduced motor defect? So that's one program that we're quite excited about. Let's see, we can move next one. I keep this advancing. Okay. I'm not able to advance this. Okay, thank you. Just shut that. Oh. Okay, great, thank you, thank you. Another area of very active interest is the gut microbiome. I told you that there is evidence that the disease may initiate in the gut. There may be some local deposition of alpha-synuclein in gut epithelial cells, and that may somehow get to the vagus nerve and project up in the brain. It's a very active area of investigation of the gut microbiome. Its composition appears to be somewhat distinct in patients with Parkinson's disease. Also, we have another investigator looking at the nasal mucosa, which has its own microbiome, to see if any of the microorganisms that may be distinctive in Parkinson's disease somehow elicit the accumulation of alpha-synuclein in the nasal mucosum or in the gut epithelium. And so there are several teams that we've invested in who are doing this one, and one in particular at Duke that I'll be visiting with actually next week. There are other programs that we've funded. We've funded a total of 163 laboratories organized into 35 teams in 80 different institutions in 14 different countries. And so these teams are asked to communicate in an online forum. We have weekly group meetings that different teams participate in. And as I said, our expectation is not just that individual laboratories publish great papers, but that they do so in collaboration with other laboratories to bring the different strengths of these teams together. So that's the current makeup of the network, and I want to leave you with the first and only in-person meeting that we had last year. We got together finally after the pandemic, and we had a meeting in a nice place in the Bahamas. This is these are all the assembled principal investigators from all over the world that were happy to join forces with us. This is these are my colleagues in the front row. To my left is Kemeny Riley. To my right is Todd Graham, who is the lead scientist at the Michael J. Fox Foundation. So, and these are all the PIs arrayed above us. So we look forward now to continuing. The funding has been going for 2.5 years. We've committed now more than $1 billion, not just to these teams, but also to coordinated efforts to collect thousands of patients and samples from these patients. and to the NIH for infrastructure, genomic infrastructure that we've invested in. And it's a big gamble. Mr. Brin is a very generous benefactor. He asks very little of us. Indeed, I had difficulty getting a meeting with him, so so this is a great gig if you could get it. $1 billion investment and no overhead on the part of the guy who's making the investment. So the gamble is that through all this effort, we will identify new targets, new cellular targets and molecular targets that our very entrepreneurial members of the network will take with them to develop IP and to find funding elsewhere to proceed with drug discovery and clinical trials. But for the time being, all of the work that we do is going to be in the public domain. Our investigators are free to patent their discoveries. We'll ask for no patent position on them. But everything that they do has to be published openly so that investigators around the world will be able to take advantage of what we learn. So fingers crossed. Thank you for your attention.
Take some questions, and he is going to facilitate his own Q&A. So,
yes.
So it seems as though in Alzheimer's disease, dysregulation of glycolysis has been related to progression of the disease.
Mm-hmm.
You've shown that this vagus nerve is also related. You know that the microbiome produces something like 80 to 90% of the dopamine in the human body. Do you think that there are potential dietary therapies that are also available for Parkinson's disease that might be related and might be able to help?
Yeah, could be. There are always claims of this, but I'm dubious of most of the claims about diet. And even in the environment, it's almost certain that there are environmental factors that potentiate the disease. But the strongest correlation with Parkinson's is an anti-correlation to cigarette smoking. This is not something that I would advise you to take up, but there it is, and there's no explanation. It's not nicotine, it's some other volatile in cigarette smoke that is anti-correlated, and of course it's been corrected for the death due to lung cancer. But in respect to glycolysis, one of our teams presented some very interesting results recently on the effect of a drug that is believed to stimulate the activity of the glycolytic enzyme phosphoglycerate kinase. And there is a correlation in the literature from the past year that suggests that men who had prostate problems, who took a drug that's no longer on the market, but which is known to stimulate PGK, had a significantly reduced incidence of Parkinson's disease while they were on this drug. So that's a fascinating correlation. It remains to be demonstrated, though, that the drug isn't doing something else. And now this particular team is actively looking for new or directed modifiers of PGK activity. And if that's true, then PGK turns out to be the rate-limiting step for ATP production and glycolysis. So if it's that simple, and all you need is to stimulate that enzyme, then that could be something that would be really powerful, that could relate to diet. Yes?
Randy, given you mentioned all these different components of the disease or different things that are correlated, from the problems with mitochondrial dysfunction or the protein kinase or synuclein, it sort of suggests that personalized medicine might be needed for each person. They won't have the same spectrum of problems, but are there even diagnostic markers for each of those categories that you showed in your slide where you can do that for a given patient to know which set of targets you need to treat in that patient versus another?
Well, not yet. The only clear diagnostic tool that's just becoming available is to measure, as I mentioned, this activity that can be detected in CSF that stimulates the aggregation of alpha-synuclein. It's been turned into a clinical diagnostic test that could be routinely used. The others, there's no routine, easily monitored marker for the activity of the LRRK2 kinase. there is a company in San Francisco called Denali that has devised a drug that's now in phase 2 trials that you can dial in an effect in reducing the LRRK2 kinase activity. In the case of the mutant allele that Mr. Brin has, it's possible to just dial it down by a factor of 2. They've shown that the drug, they've devised a drug that has easily passes the blood-brain barrier. They've shown that it engages the target, that it reduces the activity of that enzyme that they can measure in laboratory animals. They've shown through phase 1 trials that it's perfectly tolerated, and fingers crossed now on the phase 2 trial. But all the others are not easily monitored by routine clinical diagnosis just yet. But the hope is that, I mentioned just briefly, that in biopsy patients, there's a big brain bank in Arizona. And in biopsy samples that have been evaluated, both of patients who had the LRRK2 allele who died, and then patients who had the sporadic, kind of random onset form of the disease, they both have a dearth of cilia on striatal neurons. And is that just because they died for other reasons, or is it directly related to the LRRK2 effect that I mentioned? And if it's related, then the hopeful thing would be that drugs that mitigate or that reduce the activity of hyperactive LRRK2 would also have an effect in forms of the disease that are not related to LRRK2. But that's just a guess. I mean, if all that comes from the Denali drug is that it affects those patients who have the LRRK2 mutation, that'll be great. Although we don't want to discourage Mr. Brin from continuing to invest in curing this form of the disease. So it's lots of shots on goal. I think a lot of you have followed the saga of the immune therapy for Alzheimer's and these antibodies that the Biogen company has been promoting. Many of us feel that the amyloid hypothesis is a dead end that's cost the pharmaceutical industry tens of billions of dollars. So we don't want to be caught in the same trap, putting all of our eggs in the Lewy body basket. We want to look at lots of possible targets and not invest ourselves too closely in any one. Yes, Roger?
Just a follow-up question. What fraction of PD cases have a heritable factor?
It's estimated to be as much as 30%. There are many GWAS loci that have not yet been ascribed to a gene that are found in other familial forms. They may have influence regulatory pathways that turn on or off things that include the genes that are known. So, but still, the suggestion, the belief is that most of it is sporadic. Now, it could be sporadic in a genetic sense, that is not being non-familial. It could be mutation in the embryo, such as has been found for autism, or it could be some environmental toxin that's just not well understood yet. Yes.
Do we know what makes the difference between the projection for India and China?
Oh, I wouldn't be qualified to give an answer to that. My suspicion is it's a question of the diagnostic power of the two countries. I think lots of India is very rural, and there may not be as much diagnostic power in clinics in rural parts of India. But lots of China is rural as well. I really couldn't offer a good explanation for that apparent difference. Yes.
Thank you for the beautiful talk. I was wondering, do you have any thought on why the dopaminergic neurons are more vulnerable?
Yeah. Well, that one image that I showed you of the cell and its enormously complex projection of nerve terminals indicates to me that why it's so vulnerable. It is hyperactive. It's always turning on signaling. It's involved in millions of signaling events per cell over a short period of time. So you can imagine anything that compromises the production of ATP by mitochondria would be viewed as lethal to the function of the cell. And so, to me, that's the most obvious explanation. Because these mutations, LARC2 and PINK1 and Parkin, they affect the same processes in all of our cells. And yet, not all of our cells are dying at the rate dopaminergic neurons are dying in patients with these mutations.
But it's that mean different neural, other neural types outside the neural are able to
Are able to,
more able to defend this kind of
perhaps. But, you know, I would point out to you that the model that I gave for the communication between the dopaminergic neuron and the cholinergic striatal neuron suggests that the lesion, the LARC2 lesion, may actually be in the striatal cholinergic neuron, not in the dopaminergic neuron. It's that striatal neuron that appears to fail to produce cilia, which then fails to communicate with the dopaminergic neuron, which leads to the dopaminergic neuron's death. But it also leads to the cholinergic neuron's death, too. Yes?
I'm intrigued by the preserved function where you showed the Parkinson's patient playing basketball and
Yeah.
being able to ride a bicycle. Are those preserved functions always observed with highly learned habitual
Yeah.
types of behavior, and do you have a mechanism?
Yeah, you know, that's a
for
great
that?
question that I have no answer to. Is that something that anybody with Parkinson's can get on a bicycle and start riding? I don't know. It may be something that was a learned behavior from past experience that can simply be resurrected. And in the case of Ben Stetcher, you know, he's played basketball his whole life. So those movements and the dribbling between his legs is something that he obviously learned before he had the disease. So he's somehow able to resurrect this neural circuit, this so-called open loop circuit that can overcome the deficit in his dopaminergic neuron circuit. And so the hope is that those open loop circuits could be trained in individuals who may not have had that learned behavior. But that's, of course, a long way off. One more.
Is it known what the role of melanin is in those dopaminergic?
Yeah, that's another great question. We have a team that's in Spain, 5 different laboratories and different institutions in Spain who made that the focus of their investigation. What is it about melanin? And could it be that melanin is itself toxic to dopaminergic neurons that could explain the progression of the disease? I don't think there's a good answer to that. But that's definitely something we've invested in. Yes?
I'll just ask your opinion on this. So obviously there's a lot of hypotheses nowadays about the start of the onset of Parkinson's with all these competing factors that you mentioned with calcium homeostasis, copper homeostasis, misfolding, alpha-synuclein. I know you want to put your eggs in more than one basket,
Yeah.
but if you just had to pick one,
Yeah.
where would you start?
Yeah. Well, you know, for the genetic forms of the disease, they almost certainly present in different ways. In fact, the genetic forms of the disease are in some ways quite distinct from the sporadic form of the disease. I would guess, for the time being, that the most common ailment has to do with alpha-synuclein somehow. The prevalence of these Lewy bodies is pretty profound. But I'm just very cautious about that because the same would have been said about the presence of amyloid plaques in the brains of Alzheimer's patients. It's a correlation, but it is far from a perfect correlation. But that's a good place to begin, and there are drug companies, I've learned of that are active at really interesting studies on things that could be mobilized to degrade alpha-synuclein. There's a paper that was in the PNAS last year from a group at Harvard led by a guy named Bruce Spiegelman, who's been studying a hormone, a peptide hormone, that is more actively secreted during exercise. And there's also some substantial evidence that exercise may delay the progression of neurodegenerative disease. Anyway, this hormone called irisin, he showed last year when given to neurons in culture, primary neurons or cultured cells, more rapidly degrade aggregated forms of alpha-synuclein than control neurons not treated with the hormone. And he's been studying the pathway that this hormone acts through And if it actually does stimulate an autophagic process of degradation of synuclein aggregates, that could be very promising. So he started a little company, and I've got him involved with our network to test the role of irisin in a non-human primate model. So that would be very exciting. Lots of shots on goal.
So you're all invited to continue this conversation and general conviviality to reception that go out this door, turn to your left into 125. You'd be welcome to join us there. And please now join me in thanking Professor Schekman.